thank you for inviting me to. I would like to thank the organizers of the International Conference on Cancer Science and Research for inviting me to present our research. I just would like to have a pointer here first, and the title of our presentation is that Transcriptional Regulator PRDM1, Key Insights into its role in NK cell homeostasis and malignancy. My name is Küçük. I am from Dokuz University, Turkey. So different than many other people here, actually, we are working on lymphoma. As you know, most people working on cancer work on carcinomas. However, we work on different type of cancers called lymphoma. And today I will talk about the rare type of lymphoma, which is quite aggressive, called extranodal NK T cell lymphoma. So this is a rare subtype of non-Hodgkin lymphoma, and there's a geographic predilection. It's more common in East Asian populations and Latin American populations. As you know, that around 15% of the cancers have viral etiology, so there's a viral cause, and this is the case for NK lymphoma also. It has a strong association with Epstein-Barr virus. The name is NK T cell lymphoma, but most of the time these cancers arise from NK cells in 70–90% of the time. But it may also arise from cytotoxic T cells, mainly gamma delta T cells. Most of the time it's observed in males. The median age is 40 to 50 years for diagnosis. But as you can see in this picture, actually, this is a Chinese patient, a lady with NK cell lymphoma, and you can see that because of the location of the tumors which found in nasal and paranasal areas, this has also, other than the fact that it's aggressive, it's also, so it has negative consequences in terms of psychology of the patients, because face is the first place people generally look, as you know. When we look at the pathology and diagnosis in terms of histology, they are angiocentric and angiodestructive. So it means that these tumors generally destroy the blood vessels, and it is associated with high levels of necrosis as well. With respect to immunophenotype, they express generally the CD56, which is an NK cell marker, and cytotoxic markers as well, such as TIA1, granzyme B, and perforin. They are surface CD3 negative, cytoplasmic CD3 epsilon positive. EBV association can be detected by in situ hybridization, by detecting the EBV RNA called EBER. This is a rare type of lymphoma, as I mentioned. However, it's a aggressive lymphoma type. In localized stages, that is stage 1 and 2, it's not so much aggressive. However, 20% of the patients are detected at the advanced stage, which is stage 3 and 4. And as you can see from the Kaplan-Meier plots here, after 5 years, at least half of the patients pass away due to NK cell lymphoma, regardless of which kind of therapy is used. As you can see here, asparaginase-based therapies are generally give better results. And gemcitabine, if it is added also, it can improve the results, but it doesn't matter whichever therapy is used. After 5 years, almost half of the patients die if it is at the advanced stage. Now, today I will talk about PRDM1, which is a transcriptional repressor. It's the master regulator of terminal differentiation of lymphocytes. This gene is important in generation of antibody-secreting cells from B cells, and it represses the B cell identity genes such as PEX5 and BCL6. It's critical for CD8 positive T and CD4 positive T cell function, as well as terminal differentiation. It drives terminal differentiation of CD4 positive T helper cells and CD8 positive T cells. Now, PRDM1 is an important gene, and its another name is called Blimp1, B lymphocyte-induced maturation protein 1. This has been shown in vivo and published in Cancer Cell a long time ago that there's inactivating mutations. First of all, in human activated B cell type diffuse large B cell lymphoma, B lymphoma was shown to have inactivating mutations and B allelic deletions. And in vivo, using a B cell conditional B lymph1 knockout mice, when Blimp was knocked out, the ABC DLBCL type of tumors that's similar to the human DLBCL was established. In T cells also it was shown as a tumor suppressor. In anaplastic large cell lymphoma, a significant subset of patients showed deletion of PRDM1, and through in vitro and in vivo studies, it was shown as a tumor suppressor, and it was published in Blood. So when I first started my PhD, actually, there was a study in our lab that was trying to identify the common region of deletion in 6q21 to identify new tumor suppressor genes. And all the PRDM1 story actually starts from this point. So using array CGH and gene expression profiling, there was a minimal region of deletion in 6q21 was identified, and there were 3 tumor suppressor candidates here, of which one of them PRDM1, other ones are ATG5 and AIM1. So at that time we did mutation analysis, and we identified in NK cell lines, in 2 NK cell lines, mutations that inactivate PRDM1. So NK92 is a malignant NK cell line name. We identified at that time a mutation that generates a premature stop codon, thereby generating a truncated protein. In another neoplastic NK cell line, PI3 cell line, we had identified an intron mutation, which is in the intron-exon junction actually, that was leading to abnormal splice junction, so that this was also eventually leading premature stop codon formation. And in both situations, there was a truncated version of PRDM1 is found. As you know, that if there is a gene has deletion, is located in a deleted region, and if there are inactivating mutations, it is suggesting that it may be a tumor suppressor gene in that kind of cancer. And we also did methylation analysis with the cell lines. By the way, I forgot to mention that the array CGH, we had some both tumor samples and NK cell lines here available, so that during the identification of this minimal region. But we did initial analysis with the cell lines mainly, and other than deletion and mutation, we identified also promoter hypermethylation in many of the cell lines. So out of 8 NK cell lines studied, half of them had promoter hypermethylation. And as you can see here, most of the cell lines has 6q21 deletion, other than 2 of them. Even the ones that doesn't have deletion, it's not located in the deleted region, such as NK92, there was at least 1 copy has the inactivating mutation, whereas the other copy has the methylation. So when we look at the mRNA expression, as you can see, deletion by itself was decreasing the mRNA, but eventually we need to check the protein, and what we found is that other than one cell line, which has only 1 hit, there's a monolithic deletion NKYS cell line, almost all other cell lines lost the expression of PRDM1 at the protein level. So this suggested us to proceed with further studies. And what we did is that, first of all, we had identified the deletion status of the 6q21 with array CGH, which is a genomic methodology prone to false positives. So we did qPCR to validate this result. And as you can see here in NK cell lines that's supposed to have 6q21 deletion, we validated the deletion identified with array CGH by using qPCR as well. And you can see that there are gamma delta T cell lines that's not supposed to have deletion. They didn't have deletion as negative control, and tonsil was the control. In NK cell lymphoma, we extended this study and accumulated more tumor samples. Actually, as I mentioned, this is a quite rare type of lymphoma, and we had, I think, around 17 cases at that time. And this was a relatively good amount for a very rare type of cancer. And again, we did qPCR, and we showed that this deletion event was frequently observed in NK cell lymphoma tumor cases. And when we did expanded our methylation study, and when we applied BioTouch pyrosequencing to both NK cell lines and NK cell lymphoma case tumor samples, what we observed is that there was a differentially hypermethylated region in most of the cell lines as well as the tumor samples, which was not present in the control resting NK or activating NK cells samples that were actually isolated from normal human being. So promoter hypermethylation was quite frequent. Actually, we did mutation analysis as well. Unlike NK cell lines, we were not able to identify any mutation. But this was published in PNAS some time ago. Simultaneously, there was another blood publication that identified very rare inactivating mutations also in malignant NK cell lymphoma tumors. So this was all together suggesting that PRDM1 was inactivated through cooperation of genetic and epigenetic mechanisms, mainly deletion plus promoter methylation. So we did further studies to address whether it's a tumor suppressor gene or not, and we cloned PRDM1 coding sequence into a retroviral vector and transfuse one of the cell lines which doesn't have expression of this PRDM1, which is null for PRDM1. And we did JP competition assay because the retroviral vector had JP as a marker. So PRDM1 was separated by IRES, internal ribosomal entry site in the vector from JP. So JP could be used as a marker of transfused population. As you can see, the transfused population diminished after some time, progressively decreased. There was a negative selection pressure. When we did the same thing, and I need to also emphasize that when we tried some cell lines, this was quite cytotoxic, so it was difficult to establish expression, but the pattern was always clear when we ectopically express PRDM1 in NK cell lines. There was a progressive decrease in the JP positive cells in the JP competition assay, whereas there was not much difference in the empty vector transduced cells. And here in panel C, you can see the KHYG1 cells, the same ones in panel A, this time transfused with the proto-oncogene human telomerase reverse transcriptase. And as expected, actually, it's generated the opposite result with positive selection of the cells. So once we saw that PRDM1 was acting as a tumor suppressor gene, we want to do further analysis, and we did apoptosis assay with MXM5 staining. We transfused 2 cell lines, and I think after 2 days we did this assay and gated the JP positive cells for MXM5 positivity. As you can see, in both cell lines, there was an increase in the MXM5 positive cells, which is a marker of apoptosis, as you know. And we also identified that there's an increase we did also cell cycle assay with the same cell lines, and we found that compared to vector-only transduced cells, there was an increase in the PRDM1, increase in the cells in the G2M phase with a simultaneous decrease in the G0, G1, and S phases, which was suggesting that it may be inducing G2M cell cycle arrest. We didn't only do overexpression studies, so we didn't only do that way, we did the other way around also. So what we did is that we generated the PRDM1 short hairpin RNA and cloned under the miR30 backbone into a retroviral vector. This also has a JP as a marker. So E means enhanced JP, is JP. So what we did is that we first of all, we transfuse the cell line, the NKY cell line, which already has the PRDM1 expression. It is the only cell line actually that has PRDM1 expression, and we did the JP competition assay similarly. Compared to 3 days post-transduction, at day 7 days after transduction, there was an increase in the JP positive cells with PRDM1 knockdown, which was not that a similar level in the vector-only transduced cells, as you can see here. And also we use primary human cells, primary NK cells, and the transduction, but NK cells were very difficult to induce for proliferation to be used for retroviral transduction. So we use a spatial system of co-culture. We took the human peripheral blood lymphocyte and co-cultured with a spatial NK cell target cell, K562 clone 9 MB21. When we put them together, NK cells expand, proliferate, and reach to purity. And after that, we did during this time, we did transduction and knocked down PRDM1 in primary NK cells with shRNA using the same vector, and did the JP competition assay. As you can see in panel D, there was also increase in the JP positive cells in time after transduction with PRDM1 shRNA knockdown. So if we overexpress PRDM1, it was negatively selected. If we knocked down PRDM1, it was positively selected, both in NK cell lines and primary NK cells. Now I would like to talk about IL-2 signaling pathway on the role of IL-2 signaling pathway on NK cell proliferation. Actually, IL-2 signaling is quite important for NK cell activation, including proliferation and survival. And there were so many, several genes actually differentially expressed between IL-2 activated NK cells and non-activated peripheral NK cells. That was reported by Dybecker et al. In 2007. So IL-2 has binds to a receptor which has three different components: IL-2 receptor alpha, beta, and gamma. If IL-2 receptor alpha, which is CD25, is not present, the other components still form the receptor, but it has an intermediate affinity. If IL-2 receptor alpha is also present, there is a high affinity receptor forms. This IL-2 receptor alpha is important because, as I will show you later, we found this as a target of PRDM1. So IL-2 receptor alpha was shown to be upregulated during rapid NK cell proliferation in CMV infection. So this is an upregulated gene. It means that when there is a need for NK cell activation, NK cell is becoming more sensitive to actually IL-2, and it can expand better and get activated better. What also we had found that before that exogenous PRDM1 induces apoptosis more potently at low IL-2 concentrations in NK cell lines. These NK cell lines, all of them are interleukin-2 dependent, and they actually, as you can see here, from the annexin-5 positivity, when we decrease the IL-2 concentration, the annexin-5 positive cell percentage was increasing, suggesting that PRDM1 may be targeting IL-2 receptor alpha or some other component in the IL-2 signaling pathway. And when we decrease the IL-2 concentration, the apoptosis effect is more prominent. So this is the system, as I mentioned, we use if we want to work with the primary NK cells. Peripheral blood lymphocytes co-cultured with the target cells KF62, CL9, IL-21. When we co-culture together, NK cells proliferate and reach to purity, and we can use for transduction with the vector. As you can see, although co-culture system induce the proliferation, but when the target cells are totally cleared, actually this proliferation stop and cells stop dying. The reason why I'm showing is that we use this system for ChIP-seq experiment to detect actually CD25 as a target of PRDM1, as I will show you in the next slide. So PRDM1 is expressed at the protein level 10 days or 13 days here after co-culturing. And when we did ChIP-seq with these co-cultured cells, these are NK cells expanded from, these are normal primary NK cells obtained in after 13 or 14 days of co-culture. We did ChIP-seq and we identified the PRDM1 binding site in the IL-2 receptor alpha component of the IL-2 receptor. Its another name is CD25. As you can see, this was also present in the IL-2 activated peripheral blood NK cells, which is activated for 6 days. So there was a PRDM1 binding to IL-2 receptor alpha, and the PRDM1 consensus sequences were there also, GAAA, GT. We also validated this with ChIP-qPCR in co-culture cells, which is co-cultured for 14 days. That there's a binding of PRDM1 to IL-2 receptor alpha. And also the mRNA expression of CD25 IL-2 receptor alpha was decreasing in these co-culture cells, suggesting that PRDM1 was probably repressing this CD25 expression. So the next thing we did is that we used two PRDM1 null cell lines that has inactivation due to different mechanisms, as I mentioned earlier today, KHYG1 or NK92. We did retroviral transduction with empty vector or PRDM1 containing vector, then sorted the JP positive cells and performed, we used the RNA for a different type of assays, such as DNA microarray RNA sequencing and qRT-PCR to assay the IL-2 receptor alpha mRNA expression. As you can see that PRDM1 downregulated CD25 or IL-2 receptor alpha expression in both cell lines. This was also validated by RNA sequencing, the PRDM1 downregulated IL-2 receptor alpha, and also cross-validated with a locus-specific assay QRT-PCR that RNA is downregulated. We also, in the same system, we take the JP-positive cells and assay the protein expression with FACS, and we found that the CD25 expression decreased in both cell lines by PRDM1 in protein level as well. So this altogether suggested that PRDM1 may be targeting CD25 to decrease the IL-2 sensitivity. But we also did some other assays such as this one. We use a CD25 with IL-2 receptor alpha, as I mentioned, siRNA, and knock down the expression of CD25 in KHYG1 cell line, which expresses CD25 at high levels. And as you can see, there was a negative selection of the cells when there is limiting IL-2 concentrations are used. As you can see here, we use two different shRNA to knock down CD25. And when there is low IL-2 concentrations is expected, the cells couldn't grow well. So there was a negative selection pressure observed. This was also observed in this, similarly in another cell line when we knocked down CD25 with two different shRNAs in both shRNA knockdown sample groups. We observed decrease in the GFP positive populations, which was somewhat expected. And we also did the other way around, and we cloned the CD25 coding sequence into a retroviral vector, which again has GFP. And this time we use the primary NK cells that is obtained through co-culture system that I mentioned in earlier slides. And after ectopic expression of CD25 provided proliferation advantage to these cells when the IL-2 concentrations are low. So based on these results, what we propose is that if PRDM1 expression is high, is targeting CD25, which is IL-2 receptor alpha, decreasing the expression, and this leads to the formation of a low affinity IL-2 receptor complex. When there is a low affinity IL-2 receptor complex is formed, IL-2 induced NK cell expansion is also decreasing. And in the other situation, if PRDM1 is low, CD25 is high, there is a high affinity complex formed because IL-2 receptor alpha is here, and IL-2 induced NK cell expansion can happen better. So this was suggesting that PRDM1 may be inhibiting certain key components of the NK cell activation, such as IL-2 receptor alpha. So these were already published results, and now I will show some results that we haven't published yet. So we used these two cell lines again and did retroviral transduction with PRDM1 alpha or empty vector. And after 48 hours of transduction, we sorted GFP positive cells and then isolated total RNA and performed DNA microarray with affirmatic series U133+2. And then we identified the number of genes that are transcription downregulated in both cell lines or in each of the cell lines. As you can see that there was at least, based on our system, there was 93 genes that were downregulated by PRDM1 in both NK92 and KHYG1 cells, more than 1.5-fold. So when we look at the genes, the results seem to be quite interesting. That PRDM1 may have a master regulator role in actually inhibiting the NK cell activation through multiple means. For example, STAT4, STAT6 was inhibited, which is important in NK cell activation and gain of effector functions. And we identify certain very critical genes such as NKG2D, which is an NK cell activation marker, or CD160 that was also downregulated. These were all involved in NK cell activation. I mentioned already IL-2 receptor alpha, but we found other components of the cytokine receptors such as IL-18 receptor alpha. These are also important in case activation and gain of effector functions or interferon gamma secretion. So one of the interesting groups we identified was actually toll-like receptor 3, EM2, IFIT2, IFIT3. These are important in recognition of pathogen-associated molecular patterns actually, or antiviral defense. And we identify some other genes important in the antigen presentation pathway. So in conclusion, PRDM1 is inactivated by combination of genetic and epigenetic aberrancies. So PRDM1 is located in the 6q21 region and is deleted in several NK cell lines and tumor samples. As I mentioned earlier, it is inactivated mainly through monolithic deletion plus promoter hypermethylation in both NK cell lines and NK cell lymphoma tumor cases. Very likely there are some mutations, but they are quite rare, not so much common in the tumor samples. So we also showed that PRDM1 acts as a tumor suppressor gene in NK cells. It causes negative selection of NK cell lines with ectopic PRDM1, resulted in cell cycle arrest and apoptosis. Also positive selection of NK cells with PRDM1 knockdown. And PRDM1 transcription downregulates also several key genes involved in NK cell activation, including IL-2 receptor alpha and NKG2D. So with that, I would like to finish my presentation by acknowledging our lab members. Current lab members are listed here, previous lab members here. So this is our website. If you are interested, you can also check there. I'm working in Dokuz University Faculty of Medicine. And I would like to also thank the funding agencies that supported our projects. Thank you for listening.